Good morning, everyone. It's my distinct honor this morning to introduce Dr. Molly Wagster, who is the Chief of the Behavior and Systems Neuroscience Branch in the Division of Neuroscience at the National Institute on Aging. Now, Dr. Wagster received her Ph.D. in experimental psychology from Tulane University and then completed a postdoctoral fellowship in neuropathology at Johns Hopkins University School of Medicine. And for over a decade, Dr. Wagster investigated the neural mechanisms of learning and memory, changes with age in animal models, and studied changes in neuroreceptor mechanisms in relation to cognitive decline in neurodegenerative diseases such as Alzheimer's and Huntington's disease. She then joined the NIA, where she oversees administration and development of research in cognitive and emotional change with age and in sensory and motor disorders of aging. And she directly manages a portfolio of research in mechanisms of cognitive change with age that spans research from molecules to behavior. And in this context, Dr. Wagster has been an enthusiastic supporter of career development of investigators on cognitive aging, and I can attest to that because she's been my program officer since I was a postdoc, which is now 20 years ago. She served at the NIH Program Officer for the development of the NIH Toolbox and assessment of neurological and behavioral function, and currently serves as the science officer for the outreach dissemination core of the Person-Centered Assessment Resource, a trans-NIH cooperative agreement. And she's really a tireless advocate for research on cognitive aging, and she serves as the federal liaison for the AARP Global Council of Brain Health, and leads the NIA component of the Joint McKnight Brain Research Foundation NIA Research Partnership in Cognitive Aging that many of us participate in. And she was really instrumental in leading to date 3 cognitive aging summits that have brought together over 300 leaders in the cognitive aging field. The last one that was focused on issues related to cognitive resilience and reserve, which I believe we'll hear about this morning. So without further ado, Dr. Molly Wagster.
Good morning. I can hear myself, so I'm assuming everyone else can hear me well. Okay, terrific. Wonderful. And thank you, Jen, for the introduction, and I thank Mike and Manuela for inviting me to participate in this wonderful meeting. It's really been a pleasure to be here and to interact with all of the individuals and to really get to hear some of the cutting-edge science. So I'm very pleased. I have no disclosures to report. The title of my talk is Cognitive Reserve and Resilience, How Can We Do Better? And it really has sort of a double meaning. One part is that sort of how can we do better, in the sense that how can we find out through research, animal research, human research, how we may improve our ability, our prospects of maintaining brain and cognitive function as we age and become successful cognitive agers. But also, how can we do better as researchers, as federal employees who are promoting the research in order to get the work done that needs to be done and in the most efficient way. So I'm going to be illustrating some discoveries for the concepts of resilience and reserve, but also at the same time demonstrating how NIH works to establish programs of research and set research agendas. I've tried to not speak about things that have already been talked about in other plenary sessions, so I hope that I will be presenting some new things to you today. So some of the early findings leading to the conceptualization of reserve or resilience. This is a 1968 article from Blessed, Tomlinson, and Roth, in which they were looking at the relationship of mental test scores or mental exam scores against plaque counts. Of course, these are individuals who were deceased, and they were looking at the pathology in the brain. And it's really not a perfect relationship. Of course, those people who had very good cognitive performance had very low plaque counts, but there were also many people who had very good cognitive performance who had high plaque counts. And in the article, the authors stated, "It would appear that a certain amount of the change estimated by plaque counts may be accommodated within the reserve capacity of the cerebrum without causing manifest intellectual impairment." So thus introducing this notion that you can harbor pathology but still be able to perform well. 20 years later, Katzman and colleagues published this paper, again, some of the earlier findings leading to the conceptualization of reserve. This shows individuals. Okay, Mike, now I'm having a problem. Okay, here we go. In the lighter bars, individuals who are non-demented by virtue of their response on a fluency score, with and without pathology, with being the solid bar, and then those who have a diagnosis of dementia, frank dementia, with and without pathology, showing the, by again, those demented with pathology showing the solid bar. And in the lower panel, it shows plaque counts in these individuals. And I'm drawing your attention to the multiple areas of the brain that Katzman and colleagues examined, and that those with demented with pathology and non-demented with pathology are really not showing significant difference in the number of plaques, and yet these individuals differ quite significantly in terms of their cognitive performance. So these are some, one of the thought leaders in the area of, particularly of cognitive and brain reserve actually, is Jakob Stern. And these are just some of the tenets that Dr. Stern has put forward in trying to explain or wrap ourselves around the concept of reserve. That reserve may explain the disjunction between the degree of brain damage and the clinical manifestation of that damage. So in Dr. Stern's estimation, brain reserve is, the emphasis is on the brain hardware, the neurons, the synapses, that perhaps those with brain reserve or good brain reserve have more neurons or synapses to lose, maybe from the beginning, or maybe they acquire this over time, building up neurons and synapses. And that there's the idea of brain maintenance, that somehow perhaps direct effects of lifestyle or other activities can have an impact on brain reserve. Cognitive reserve he views as more of the software of the brain, and that resilience or plasticity of cognitive networks and how they stand up in the face of disruption. Jen mentioned that we've sponsored, in conjunction with the McKnight Brain Research Foundation now, 3 cognitive aging summits. These are convened by the NIA and made possible through a generous grant by the McKnight Brain Research Foundation to the Foundation for NIH, allowing us to have a public-private partnership. The most recent summit was held just this past year, really just almost exactly a year ago, and was comprised of 6 sessions, all focused on the notions of resilience and reserve. Session 1 was how do we operationalize brain reserve, cognitive reserve, resilience, and compensation. And as it turns out, the things that were discussed in this first session, the definitions, the meanings, the conceptualization, were echoed throughout the entire summit. Each session seemed to come back to this issue of, do we really know what we're talking about? Are we all talking about the same thing? And how can we compare our data if we're defining these things such as reserve and resilience, compensation in different ways? Again, there were the other sessions were what are the threats to successful brain and cognitive aging, early life contributions to reserve and resilience, later life contributions to reserve and resilience. How do we validate approaches that aim to harness reserve to improve the aging brain? And I think this was it sounds sort of like a dull session, but it was a particularly important one, and it actually was an extremely interesting one. Is that, again, it's not exactly the same as operationalizing these terms, but how do we actually go in and measure reserve, and how do we measure resilience above and beyond defining it? And then we had a session on some innovative approaches. So there were many recommendations that came out of this summit. This was held specifically to generate recommendations, and I'm showing some here. Many of them called for what I would call resource development, building infrastructure to share data and biological materials across multiple cohorts, even establishing a registry of exceptional cognitive agers, which was of great interest to the people at the meeting, and I'm going to go into quite a bit of detail this morning about the exceptional cognitive agers. Supporting longitudinal studies of rats, allowing us to follow a short-lived animal model over time and sort of begin to explore where in the trajectory some of these changes may occur. And then, importantly, establishing common scientific nomenclature for the terms resilience and reserve. So there are many definitions, many concepts. You've, I'm sure, encountered them, maybe used them yourself. There's compensation, brain reserve, cognitive reserve, resilience, neuronal reserve, neural efficiency. There is the definitions of resilience to Alzheimer's disease. It means coping with pathology, but resistance to Alzheimer's disease means actually avoiding pathology. So we were lucky enough this year, actually in 2017, to publish a funding opportunity announcement to bring in or to establish a collaboratory using a particular mechanism at NIH called the R24 to bring together individuals to work on coming to some kind of uniform set of research definitions for cognitive reserve and resilience to Alzheimer's disease. Have to be resilient to something, and in this case, resilience to Alzheimer's disease. The deadline has already passed for submission. We have the applications in and they will be reviewed soon, but the intent of this will, once the award is made, is that the recipients of the award will be bringing together everyone in the universe, nationally and internationally, to work on these concepts and definitions and to bring something uniform to the table. All right, now I'm going to switch over to other areas in which we're working and supporting research and talk a bit about the cognitive superagers or exceptional cognitive agers. And I want to show a short video. Before I start, this was a group at Northwestern University in Chicago where they have one of the cohorts of cognitive superagers being studied in the US. This was local TV coverage of a cocktail party bringing the cognitive superagers together just about a year ago.
It is a gathering made up of seniors.
Is that loud enough?
Any seniors, they are part of an elite group of people over 80 that
Can
are
you
being
increase the
studied
sound?
because they are so-called superagers. Neuroscientist Emily Rogowski is a lead investigator.
Individuals who are over age 80 who have memory performance at least as good as individuals in their 50s and 60s.
So what's their secret? Well, that's the great mystery Northwestern researchers are trying to unravel. Why are these seniors aging at a healthier pace than their counterparts? Ask a superager and you might get different answers. Lillian Rubenstein, 93 years old.
And my approach is you've got to live life to the fullest, the good and the bad.
Herma Goldstein, 104.
Well, I go to exercise class once 5 times a week when the weather is good.
To get to the bottom of their youthful nature, scientists are focusing on the superager's brain.
This part is thicker.
And it turns out their brains display marked differences from their peers, including physical shape and shrinkage typically found in aging. And there's a particular region of the brain called the anterior cingulate cortex. That was much thicker than an average 80-year-old and closer to a 50-year-old brain.
We know that the anterior cingulate is important for attention, and attention supports memory.
So how can we all become superagers? Well, there's no definitive answers, but there are some anecdotal results from these special seniors that we could learn from. They are socially engaged and active with family, friends, and their community. They come from all walks of life, and many have suffered hard times, battling cancer, losing children, but throughout the hardships, they are very resilient and always seem to bounce back.
And I think that's something that we can kind of take home with us today to say, well, we're all going to experience stress, but our reaction to it may be important for our biology. It may be important for aging.
For these seniors, being part of this elite group is a badge of honor, and they're proud to be associated with each other. Edith Smith is 102 years old and says until recently, she was still driving on Chicago freeways. She believes to keep your mind sharp, you have to stay active and take care of yourself first.
That's why I'm a super ager.
All right. What defines an exceptional cognitive ager? Well, there are two groups here in the U.S. Studying exceptional cognitive agers, at least, again, to my knowledge. At Northwestern University, their criteria is individuals who are age 80 or over age 80 who have performance on the Ray verbal auditory learning task of episodic memory that is at least average according to 50 to 65-year-old norms. So some, 20 to 30 year, that performance of some 20 to 30 years younger than their age. And that performance in other cognitive domains such as language is at least average for age. There also is a small group of cognitive or exceptional cognitive agers that have been assembled at Mass General in Boston. These are a bit younger that they are studying, 60 to 80-year-olds. Their performance on the California verbal learning task, long delay free recall, is supposed to be at or above normative values for young adults, so in that 80 to 32-year-old range. And performance on trail making tests, no lower than a standard deviation below norms for the age. So even in this situation, we have slightly different definitions, but clearly they are individuals who are performing, either maintaining function, performing much better than individuals who are age peers. This is just some information on demographics, courtesy of Emily Rogalski. I'm just showing here very quickly what I was just speaking about, where we have cognitively average 80-year-old norms at this level on the Ray Vault, and then cognitively average 50 to 60-year-old norms shown here. And clearly the superagers are above or equal to that of individuals who are much younger than their 80-plus years of age. At the time of this slide, the group demographics, they were about 75 or 74 individuals enrolled in the super aging cohort at Northwestern. Now there's a little over 75. Their mean age is around 83. Mean education level is 16, so they're on the higher education range, but the range is from 12 to 20 years. The cohort is diverse, but not extremely diverse. There's about 8% African American in the cohort, and the majority of them are right-handed. I'll also say that most of them, not all, but most do not carry the ApoE4 allele. Some do. They range in terms of whether they were ever smokers, and some were never smokers, some were smoked but quit, and some still continue to smoke to this day. Their engagement in things such as exercise was also quite varied, and Emily has told me that some of the individuals, when interviewed, say that they run only if chased. All right, this work from Tamara Geffen and others at the Northwestern group just illustrates maintenance of episodic memory over an 18-month period in cognitive superagers. This, again, is the score on the Ray Vault. You see visit 1 at baseline, and then a year and a half later, only one individual dropped down just a bit below that range of average to superior performance. And I was also told by Emily Rogalski that at a later date that individual bumped back up into the range. And they weren't very far below at that point. So what makes them so good? Psychosocial factors, overall brain structure, circular cell level differences, resistance to normal aging brain changes, resistance to neuropathology. This is a study, again, from the Northwestern group looking at personality profiles of the cognitive superagers. What they found were on these 5 dimensions that they were low in neuroticism, high in extroversion, and high in agreeableness. There has been some work by comparison showing that individuals at risk for Alzheimer's disease tend to be high in conscientious. If you're at less risk for Alzheimer's disease if you are high in conscientiousness and low in neuroticism. So a little different, but not particularly. They also very recently published a paper looking at psychological well-being in cognitive superagers via using the Psychological Well-Being, or PWB-42 survey. On these 6 dimensions, the one that the superagers endorsed significantly above the others was positive relations. And again, this really sort of social type of thing, satisfying, warm, trusting, high-quality relationships with others. I know I can trust my friends, and I know they can trust me. Overall brain structure. You saw this image briefly in the video. This is, again, work from the Northwestern group. This image compares the brain of individuals who are average older adults, average 83, cognitively average older adults, average age of 83, versus cognitively average middle-aged adults whose age is around average of about 58. And if I can get this to work, you can appreciate by the warm colors on the surface, lateral surface of the brain here, that there are any number of areas in which the older adults show thinning cortex, less cortical volume than the average middle-aged individuals represented here. All right, now the superagers. Again, these are individuals whose average age is about 84 versus the cognitively average middle-aged individuals, same group, average age of about 58. And what do you see? There's really no difference in terms of cortical thickness between these two groups, even though one is some 20 to 30 years older than the other. Now, this work comes to us from the Mass General group led by Brad Dickerson, looking in their cognitive superager group and looking at brain structure. They looked at a number of cortical areas, as you can see across the bottom of the slide here. And just to illustrate on the side, so blown up just a little bit to see better, in most instances, in all instances, first of all, the superagers had greater cortical thickness than their age peers, the cognitive superagers. And in many cases, and illustrated here in the angular gyrus and middle cingulate cortex, the superagers' cortical thickness was that of the young adults, or comparable to that of young adults. In the same study, they also looked at hippocampal volume versus memory performance in the cognitive superagers. Here on the far right panel, this is represented by this individual who is a cognitive superager, looking at hippocampal volume, and you can appreciate, compared to this individual, that the who is not a superager but of age match, that there have much decreased hippocampal volume, and that there was a significant relationship between memory performance and hippocampal volume. So cell or circuit level differences. So just to introduce, before I get to the actual study that was done at Northwestern, I want to introduce von Economo neurons. Many of you probably know about these, but just in case you're not familiar with them, they also are referred to as spindle neurons or VENs. We see von Economo neurons in humans and non-human primates largely, but also they've been found in humpback whales and elephants. What do we know about them? Well, in humans and non-human primates, they are found primarily in cingulate cortex, but there has also been detection of them in frontal insular cortex and dorsolateral prefrontal cortex. They're large neurons, but they're rather sparsely distributed in layer 5 projection neurons. And they are lost or abnormal in autism, schizophrenia, bipolar disorder, frontotemporal dementia, all neuropsychiatric diseases in which social cognition is markedly affected. So they're thought to be, although we do not have enormous direct evidence at this point, that they're involved in emotional regulation, goal-directed behaviors, empathy, other things such as social wellness and social control. Now, Here's some von Economo paper that showed von Economo neuron counts in cognitive superagers. It turned out when they were looking in the pathology cases that had come to autopsy at Northwestern, looking at the cingulate cortex, and all of the areas represented here are portions of the cingulate cortex represented by the Brodmann areas. They noticed that there were a number of von Economo neurons in this area, and as it turns out, with superagers in sort of maroon or purple, the elderly controls in the blue-gray, and in this case comparing them to people with amnestic MCI in the green, you can appreciate that the mean von Economo neuron counts, as well as the ratio of von Economo neurons to total neurons in these areas of cingulate cortex, is much greater in the individuals who are cognitive superagers. Also, the Northwestern group examined in postmortem tissue, looking at acetylcholinesterase positive neurons, and found changes across age and cognitive performance. So here, this particular graph represents density of acetylcholinesterase positive cortical pyramidal neurons, young adults in the solid black bar, the cognitive average elderly in the hatched bar, and the cognitive superior elderly in the light or open bars. And they looked at multiple cortical regions, and I'm sure you can already appreciate the pattern that the superagers showed a lower density of these ACHE positive neurons in all of the cortical areas examined. It's not clear exactly what this means, but it is of quite a bit of interest in that acetylcholinesterase, of course, breaks down acetylcholine. And if the cognitive superagers, for example, have less breakdown of acetylcholine, that means that there is probably more acetylcholine, an important neurotransmitter for learning and memory, available and could be contributing to their superior cognitive performance. It's not known whether this is a plastic response or an adaptive response in terms of the diminution of acetylcholinesterase density, positive neuron density over time, or whether this is something that, for example, they were born with. Resistance to normal aging brain changes. Again, from the Northwestern group, this is showing an 18-month change in cortical volume. You can see that the superagers seem to be rather resistant compared to the cognitively average 80-year-olds, showing about a little over a 1% change over a 1.5-year period, with the cognitively average 80-year-olds, their age peers, showing much greater or well over 2% change during that same period. Resistance to neuropathological changes. This is, again, work coming out of Northwestern, looking at mean neurofibrillary tangle counts and amyloid plaque counts. Again, comparing superagers to elderly controls to people with amnestic MCI. Again, I think you can appreciate that the superagers are showing much less pathology in the brain regarding this amyloid type of pathology, neurofibrillary tangles or amyloid plaques, with, of course, those with amnestic MCI showing the greatest. They still have plaques and tangles in the brain, and some of them have it that it would be at the level that would be diagnostic for Alzheimer's disease, at least neuropathologically. I'm talking about the cognitive superagers, but it's far less in these. You see these pathologies far less in these individuals. This is all in cingulate cortex at this point. Now I'm going to switch to another group. This comes from Claudia Kawas's 90+ study. Dr. Changiz Gyula from Northwestern has been working with Dr. Kawas's group to study some of the 90+ participants with superior cognitive performance. This panel on the right-hand side shows the different stages of pathology, the Brock stages of pathology, all the way from 1, with basically no pathology, to stage 5 and 6, where you get the greatest pathology and would be indicative of a diagnosis of Alzheimer's disease. So despite similar and superior memory performance, the participants in this study who had superior cognitive performance displayed divergent patterns of Alzheimer's pathology. Some of them had the pathology, some of them didn't, similar to the Northwestern group that I was just describing. In the 8 cognitively superior participants that they identified, there was a density and distribution of neurofibrillary tangles ranging from Brock stage 1 all the way to Brock stage 6. And 2 of the participants satisfied the pathological diagnosis of Alzheimer's with Brock stages 5 and 6. And yet they had superior cognitive performance. Now, very interesting to me, and remind everyone in the room that these are based on very small n's. But this is extremely compelling to me, and one of the reasons I hope that we get a chance to pursue some of these studies in the manner I'm about to describe. These are neuronal counts in the CA1 region of the hippocampus. You'll see, I'm not very good with this pointer. I guess I need remedial training. These are 90-plus participants with Brock stages of the superior cognitive performance, with Brock stages 1 to 3. Here are the 90-plus participants with neuropathology sufficient to warrant a pathological diagnosis of AD, compared to those who have frank Alzheimer's disease, a confirmed diagnosis of Alzheimer's disease. And you can appreciate that really the neuronal counts between those with superior cognitive performance, despite their heavy pathology load, is not too different, but certainly different from those with Alzheimer's disease. And you see a similar pattern in frontal cortex. Again, those participants with superior cognitive performance, yet having significant neuropathology in terms of plaques and tangles, really showing not that much diminution of neuronal counts in the frontal cortex, but certainly much more, certainly those with confirmed AD having much lower neuronal counts. All right, so recommendations from the Cognitive Aging Summit 3. Again, as I mentioned before, there are a lot of recommendations calling for resource development, particularly infrastructure that would allow us to share materials across different groups, and perhaps even to establish a registry of exceptional cognitive agers. I think these are important things that we at the NIH work with to try to push forward, because all of this, in my opinion, again, compelling data that I just presented is based, as I said, on very small n. And so how can we increase the yield from these studies? How can we increase the number of individuals that we are following? And I think we can probably only do that if we start to pull groups together and try to figure out ways to support research that would allow people to increase the number of these cognitive superagers, maybe having multiple sites across the United States and internationally. Now, this was presented yesterday in depth by Risa Sperling, and I don't want to spend a lot of time on it, but I do feel compelled to mention it to make another point. This is a sort of composite slide addressing this issue of hippocampal hyperactivation. And so, as Dr. Sperling said in her really just outstanding talk, she was talking about the fact that we'll just illustrate it actually with this panel here from Brad Dickerson, one of her colleagues at Mass General. So in this first panel here, you see the regions of interest being identified in the hippocampus. This is from a control brain, and this is showing fMRI activation maps overlaid on a structural scan. You can appreciate that in the individual with mild cognitive impairment that the activation is much greater, even bilaterally in the hippocampus, whereas in Alzheimer's disease, you really see little activation, or certainly hypoactivation. And when this was originally discovered, as Dr. Sperling said, this hyperactivation in the individuals with mild cognitive impairment was seen as probably a compensatory mechanism, that somehow they were ramping up the hippocampus to maintain good cognitive function, but that as the disease progressed, it just sort of burned out and you became in a hypoactive state. That is, again, is no longer how this is viewed, and I think in large part it has been punctuated by work done from Michaela Gallagher's lab and others. I know Carol Barnes and John Disterhoff have done work in this area that have identified that there is hippocampal hyperactivation in old cognitively impaired rats compared to old cognitively unimpaired rats. Seen here in, at least in CA3, this is a comparison of young to old cognitively impaired rats, but you would see that if this panel had also shown the cognitively unimpaired rats, they would look much more like the young. And then these are studies on the right side of looking at individuals with mild cognitive impairment, showing that in individual and administering the anti-seizure drug levetiracetam, which would reduce hippocampal hyperactivity. It did, and it also improved cognitive performance in individuals with amnestic MCI. This is work by Backer and also Mike Yassa, Craig Stark, Marilyn Albert, and others at the time from Johns Hopkins. So it indicates, I think, that if hyperactivity were something that were beneficial or compensatory, then you would think that if you were to reduce that hyperactivity, it would impair their cognitive performance. But in fact, in this case, it improves their cognitive performance and is also capable of diminishing the hyperactivity. So again, going back to the recommendations from the Cognitive Aging Summit, again, infrastructure, trying to get uniform nomenclature for these terms, establishing a registry of exceptional cognitive agers, and supporting longitudinal studies in rats. This may be our one chance, or at least one of our few chances, to follow over time organisms that without sacrificing along the way, in order to be able to see where some of these things may develop during the lifetime and identify for us and what they are, that changes in the brain that maybe precede the behavioral change or vice versa. I doubt that we're ever going to be able to, I know we will never be able to do this in humans. I doubt that we will ever really be able to do it in non-human primates that just simply live too long, even some of the smaller ones like marmosets. But I think that there is real opportunity here for performing some of these studies in rats in a longitudinal way. So as I near the end, I just wanted to indicate to you that as at the NIH, NIA, people like myself, program officers, yes, we help you with your, we answer your questions regarding applications. We listen to you when you rant about the review that you got and your summary statement. We offer what we hope is useful advice, for the next round. We monitor the progress of your grants, but we do other things as well. And one of the things that we do, and which quite frankly occupies a lot of our time, is trying to set research agendas and make sure that areas that are worthy of development get moved forward, identifying gap areas and trying to push forward on them. And I just wanted to bring to your attention that already at the NIA, and largely because of the increased funding we have enjoyed over the last 3 years and a little bit before that, but certainly significant amounts of money directed toward Alzheimer's disease research, we've been able to issue a number of funding opportunity announcements. And I just list a few here that I feel are directed toward this concept of resilience. The first two are RFAs. They already have the submission dates are already over, but as you can see, it's an unusual situation to release an RFA again. These are usually one-time opportunities, but this one was released a second time, which is interdisciplinary research to understand complex biology of resistance to Alzheimer's disease, and it might be issued a third time, we don't know. Others include major opportunities for research in epidemiology of Alzheimer's disease and resilience. And then I also listed a couple that are looking at ApoE2, focusing on that. ApoE2 believed to be a perhaps protective allele for Alzheimer's disease, against Alzheimer's disease and cognitive impairment. But these just illustrate a small number, really, of the funding opportunity announcements that we have released over the last 3 years. I counted the other night in preparation for this talk a little over 65 active funding opportunity announcements that we have out here, that NIA has out right now to promote Alzheimer's disease research. And I want to emphasize to you that when you're studying things such as reserve and resilience and trajectories that may show us the difference between successful aging and unsuccessful aging, cognitive impairment or not, resilience and reserve, that that type of research can also be couched in the area of AD relevance. And I strongly encourage you, if you are interested in these areas, to talk to program officers at the NIA about how you might structure your work and how you might take advantage of these opportunities. So this is my last one about funding, just very recently released, and this may be of interest to some of you, was a funding, well, it's an opportunity. It was released as a notice for administrative supplements for NIH grants that are not focused on Alzheimer's disease, to allow you to add some component of Alzheimer's disease to your work or something related to it. And we did this last year, NIA alone, but I want you to appreciate that this year there are many institutes who are participating in this. So, for example, if you are funded by the National Institute on Drug Abuse, and your project has something that might lend itself to an exploration through a supplement of looking at something related to Alzheimer's disease, you would qualify. And so I want to make you aware of this opportunity going forward. So in closing, I say that I work for you. I work for you as individual investigators in trying to get the research funded and advancements made and bettering the public good. But when I say I work for you, I also mean that I'm working for the public good. And together, when we're funding your research, when you're working on these issues, when you're trying to help us understand better how to avoid Alzheimer's disease, how to maintain brain and cognitive function as we age, how to become resilient, we're working together, and we're working together to promote public health through biomedical research. It's an important point not to be missed, that we're all working together on this. And I appreciate very much the opportunity to have the time to talk with you this morning. Thank you.